Hi guys, this week we're going to be talking about the genetics of cancer. So we're going to talk a little bit. Cancer has been around and has been part of our life and the life of human beings for quite a long time. History tells us that in 300 BC, Hippocrates actually coined the term cancer because he used it to describe a crab-like shape of a tumor that was invading normal tissue. Cancer has or will affect 1 in 3 people. It is genetic, but that does not necessarily mean that it is inherited. Cancer generally arises from genes that have alterations, and there are faulty instructions within those altered genes. The genetic component of cancer development is at the cellular level within the mitosis or the division of the cell. And cancer happens or develops over time. And we always need to remember that cancer is a multifactorial disorder. So some terms to keep in mind as we go forward. A carcinogen is a substance that causes cancer or leads to mutations or damages the DNA of the cell. Cancer is actually the term we use to define the disease that is caused by a loss of cell control. And so what we mean by this is that the cell escapes normal control over its division rate. And so because of this, there is a growth of tissue, and we call that tissue the tumor. A tumor can either be benign or malignant, and that has to do with whether or not there is invading to other tissues and what we see upon cellular pathological changes within that. And we know that metastasis is the spread of the tumor or the cancer from one part of the body to another, and this occurs via the blood or the lymph vessels. So there are 2 specific types of cancer-causing genes. We have oncogenes, and we have tumor suppressor genes. So when oncogenes are activated, they cause cancer to develop. And when tumor suppressor genes have an alteration, cells neglect to go through their normal cell cycle control or checkpoints. There is cell cycle control. This we mean by this is the timing, the rate, or the number of cell divisions in a particular cell are dependent upon protein growth factors, signaling of molecules outside of the cell, and transcription factors within the cell. There are checkpoints at all parts of this cell cycle control, and they ensure that in the meiotic or the mitosis that there is a correction that takes place with it if there is an alteration in any of these areas. So this is a schematic of the different type of cell cycle controls that there are within our body and within our cells. So this is in your book, figure 18.2. Just take a look at it. So you can see that there are many places within the cell cycle that alterations can occur or things can get or spin out of control. So there is a loss of cell cycle control. Many types of cancer result from faulty checkpoints. The cancer cells spread down a pathway where there is unrestricted cell division, which means these cells don't have to go through these checkpoints. Cancer cells can either lose specialization or they never really specialize. And this again visually kind of shows you how a cancer cell could develop. There is inherited versus sporadic cancer. Somatic mutations occur sporadically. They result in a single dominant mutation or 2 recessive mutations of the same gene. This increases cancer susceptibility. There are germline mutations, and cancer susceptibility is passed on to the offspring, and it requires generally more than 1 somatic mutation to occur. It's rare, but it does occur. So this is an example of if, let's say, you have lung cancer and what a germline or an inherited type of lung cancer might look like, or sporadic cancer. So we can develop lung cancer because we're exposed to carcinogens and we have this faulty regulation of our cell division related to this exposure, which causes the mutation to occur. Or we can inherit a faulty checkpoint that is enhanced or activated when exposed to a cancer-causing agent. So here is some more information for you to just kind of take a look at and read. It is in your book. It is a good explanation about how cancer begins and how if it's not halted, it can spread throughout our body. So some characteristics of cancer cells. They continue to divide, but they divide at a quicker than normal rate than our average cells. Most have an inheritable mutation. They can be transplantable, which means they can go to different organs. They are de-differentiated, which means that these cells lose their special identity, like if they're a liver cell or a lung cell or a red blood cell or white blood cell, they don't have that differentiated identity that attaches to the cell. They appear different and they look different, and they do have different types and or numbers of antigens on them. They also lack inhibition. They can induce angiogenesis within blood vessels. They can be invasive, which means they're going to squeeze into any available tissue space. And they have the ability to metastasize, which means they can move to a new location and start the process of this rapid cell division and tumor growth in that new space. Origins of cancer cells. Cancer begins at the cellular level, and usually in one of four ways. There is activation of the stem cell. There is this de-differentiation of the cell. There's an increase in proportion of tissue that consists of stem cells and these progenitor cells, or there is faulty tissue repair. Cancer lost by specialization. Specialized cells lose some of their distinguishing features as the mutations occur as they divide through the mitosis process. This is what results in what we call a de-differentiation. And so what happens is they have these de-differentiated cells and they're stimulated to reproduce more of these de-differentiated cells. There is a lot of uncontrolled tissue repair. So you can have an acute injury or a chronic injury. And this is how normal tissue repair occurs. And over on the other side is what happens when cancer, this uncontrolled tissue repair occurs and then cancer cells develop. Oncogenes. There's proto-oncogenes, which are the normal version of genes that promote cell division. Expression at the wrong time or in a wrong cell leads to inappropriate cell division and the development of cancer. Proto-oncogenes are called oncogenes in their mutated form. So proto-oncogene is a mutated oncogene. One copy of the oncogene mutation is sufficient to cause this proto-oncogene cell division. Oncogenes are activated when proto-oncogenes move to the next gene. The gene pair is transcribed together, and this creates this fusible protein, which then either activates or lifts the control of cell division, which leads to the rapid growth and division of cancer cells. So here's a picture of what it might look like if an oncogene or overexpression of normal function. So we have acute promyelocytic leukemia. If you develop this disease, there is a translocation between chromosome 15 and 17. The combination of the retinotic acid cells, surface receptors, and an oncogene, there develops a fusion protein functions as the transcription factor. When this is overexpressed, the patient develops acute promyelocytic leukemia. And for some of these patients, they may respond to retinoid drugs. There is CML. Most of these patients' deformity occur at a translocation of what we call the Philadelphia chromosome, which is the tip of 9 on 22 or 22. When you have this abnormality, you produce a fusion protein. And this causes excessively to stimulate cell division. By understanding how chronic myelogenous leukemia develops and understanding where it's located has actually allowed companies to develop this new drug, Gleevec, which helps in the treatment of this disease. So here is a schematic of how that looks like in picture form. So HER2/neu is a product of an oncogene. Excessive levels are found in 25% of cancer patients. They may have too many receptors. They have too many signals, which tell them to divide. And this, if this is found, then monoclonal antibody drugs can help bind to the receptors, and this will help block the signal to divide. Herceptin is a drug that works in this particular fashion. So then we have tumor suppressor genes. Cancer can be caused by loss of the ability for your normal cells to inhibit cell division. Tumor suppressor cells normally stop the cell from dividing, so they notice the abnormality and they stop the division process. Mutation in both copies of the tumor suppressor genes is, for the most part, required to allow for this abnormal cell division. These genes can be lost, the ability to suppress the tumor, by either there's a deletion or a silencing by a promoter. So the gene is the instructions are either deleted or silenced. So retinoblastoma, which is a childhood cancer, which is located on chromosome 13. When a patient develops this, they cannot, their genes are unable to activate the genes that carry out mitosis, which normally halt the cell division in the G1 phase of mitosis. So this particular cancer is what they call a two-hit hypothesis cancer causation. So they're believing that it happens in two different ways. So the two-hit hypothesis is that there are two mutations or deletions that are required, one in each copy of the gene. For sporadic cases, which are considered non-inherited, retinoblastoma is the result of two somatic mutations. So the individual in the inherited has one germline mutation or allele in each of their cells. And because of this, they develop this particular cancer. We have the P53 gene, which is the guardian, named the guardian of the genome. This gene can determine if cells have damaged or the DNA needs to be repaired. If this P53 is damaged, it will induce, if they notice a damage, it will induce apoptosis. In more than 50% of cancers, there is an abnormality of this P53 gene. So it cannot necessarily pick up that there is a cancer or a damaged DNA, and so it does not induce this apoptosis that would normally occur. Rare inherited mutations of the P53 gene can cause certain diagnoses, certain particular syndromes, and that would cause people to develop cancer at very early ages. This is a schematic, so we can see the environmental trigger, the gene, where some of the mutations occur, or where you might see a somatic mutation occur. Breast cancer. There are two main forms. There's familial and sporadic. And we know that there's a susceptibility within the BRCA1 and BRCA2 genes, and these encode for proteins that join two other and form a complex that allows repair of the double-stranded DNA breaks. With these mutations, the genes cannot have different incidences in different populations. Inheriting the BRCA mutation increases the risk of other types of cancers as well. So MicroRNAs normally control the expression of proto-oncogenes and tumor suppressor genes. Thus, when they are mutated or expressed differently, cancer can result. Patterns of microRNA expression changes cancer processes. So we have gatekeeper genes, which directly control mitosis and apoptosis. We have caretaker genes, which control mutation rates and may have overall effect when a mutation is destabilizing within the genome. Most cancers are the accumulation of a series of several mutations within several genes. You can develop familial Adenomatous Polyposis, which is inherited familial colon cancer. 1 in 5,000 people have this, and they develop multiple polyps at an early age. Several mutations contribute to this, and you can see what they are. So there are several scale projects that are being done for analyzing genomes of cancer cells. And many of the mutations that accompany cancer interact in only a few pathways. So once that pathway is implicated, scientists are now looking for ways to develop drugs that actually target the pathway. There are environmental causes of cancer. So our environment contribute to cancer by mutating or altering the expression of the gene that control the cell cycles, apoptosis, or DNA repair. Inheriting a susceptibility puts you at further risk for developing cancer. We know that individuals can lower their chances of developing cancer by avoiding high-risk environments. And sometimes we can take chemopreventive nutrients, folic acid and vitamin D are just an example of a few. So this is how vegetables can lower your risk of cancer. You can see how it changes. We got a burger and we got Brussels sprouts. So how do we study cancer for environmental leaks? We look at population studies, cause control, case control studies, and prospective studies. Cancer diagnosis and treatment. Most often cancer is discovered on a screening test. The oldest form of treatment is actually surgery, which removes the tumor. We can use radiation or chemotherapy, which non-selectively destroys the rapidly dividing cells. We can also use drugs that do the same. There are some new types of drugs which are used to simulate cells to regain their specialized characteristics. Some to inhibit telomeres at the telomeres, to induce apoptosis or inhibit angiogenesis. So genomic information is being used increasingly, which will enable physicians' genomic information to match the patient-specific cancer to the more patient-specific treatment. So here are some, for breast cancer, some ways to how we've evolved in treatment. We used to just remove. Now we used to use phenotype to select a drug. Then we moved to genotype. Now we're looking at the genomic level to help further specialize how we treat breast cancer.